Okay, happy Wednesday morning. We're again meeting for our weekly review session. And this week, what we're doing, we're really kind of continuing our journey that we started last week and that will be on for the rest of the semester, which is looking at the nervous system of the human body. And this week in particular, we're focusing on the central nervous system. So we're looking at structures such as your brain and your spinal cord. Your actual lecture material goes into a little bit more detail than what I'm planning to cover here today, but we'll make sure that we at least have a look of some of the key things that I really want you to take away from this material. I want to start again, as I always start, with a really quick check-in, how we're doing. We'll talk about the plan for the week, and we'll also review, like I said, the key aspects that I want you to take away from this week's material. I anticipate that we will have time to quickly look at some of the examples of the weekly homework as well. And as always, I want to make time and space for any questions if you're joining this session asynchronously later on through a recording. Do remember that I'm here to help you every step of the way, so you can totally message me anytime at all, and we'll figure out answers to any questions that you might have. So starting this week, as always, I want to start with just a brief moment of self-reflection. And what we're looking at Young Pueblo's book called Inward. And since I kind of anticipated that it might be just me here today, what I've done, I've picked a number for us at random. And the number that I ended up picking was 198. And on a page 198, the thought that we're shared is going along the lines as follows. Peaceful minds have the power to create a peaceful world. And I think that the reason why this quote is so particularly appropriate for this week really has to do with the fact that what we spoke earlier on, that you're really kind of a creation of your mind. Even though we live in a very real physical reality, everything that we sense, everything that we experience about that physical reality is still a creation of our mind. So I think that that thought ties in nicely with that kind of a concept that we kept seeing. Why nervous system is so important, and why in particular we want to focus on the central nervous system. So having a look real quickly of the material that I'm asking you to do this week as a homework, I'm asking you really to focus on chapter 14. So we're doing a quiz and we're doing a practice anatomy. Both of these are due at the end of the week. I know that I listed on our material that we would also kind of venture to have a look of chapter 13. I think these chapters and these modules have a little bit of overlap. So even though the emphasis is heavily on 14, I'll pull up some themes from chapter 13 as well, and we'll take it from there. Any questions that come up, you might find that some of those answers are covered also on our material for the next week. So I think that this week and next week in particular have a lot of overlap. So this next slide that we're seeing here really kind of, I think, reminds us of the concept and of the theme that we saw, that we interact with the rest of the world and we perceive things from the rest of the world. But really, all of those are creations in our mind when it comes to giving meanings and forming perceptions on things. So I think that that's an interesting way of looking at the importance of nervous system. So I really just included this slide to just kind of bring us back there to that thought that we saw last time. kind of focusing on today and this week, we're really focusing on the central nervous system. So central nervous system is going to cover our brain and our spinal cord. And really, if you think about our brain, if you think about our spinal cord, I think especially the brain, it is just basically, if you want it to be really overly simplistic, a lump of fatty tissue. Yet it is everything you are. It is your personality, it is your memories, dreams, imagination, wishes, future hopes. So we're really quite an amazing structure that we're looking at. So I think that what we want to recognize is that not only we do all have these similar structures, but we have highly varied experiences of the world, unique experiences of the world. In addition, I think that another thing that we end up seeing is that considering that the structures are so similar, how do we end up being so different? But also as a species, how did we end up to what we are from the evolutionary standpoint of the structures that we started as? So let's have a look of that a little bit. So nearly all animals have some sort of a nervous system, whether we're talking about earthworms or going to highly evolved species like dolphins, who have very fascinating brain structures. Nearly all animals we end up noticing that they have a nervous system. And in most animals, when we look at animals, they have brains and they have similar parts, similar regions of the brain that we see when we compare these animals. Our focus is going to be largely, obviously, on the human. We will kind of compare human as well to the sheep. And I think that in my face-to-face classes, we would dissect the sheep brain. I think I've offered for you a recording where I go through the different parts of the sheep brain through a dissection activity. So we'll get to do a little bit of comparing and contrasting. How do our brains compare to that of the sheep as well? So I think that that's always useful and educational and helpful. If we look at the evolution of the mankind, I think that that's a good place to start at. One of the things that we'll end up noticing that is that over the past 3 million years or so, the human skull has grown quite significantly in the size. But another thing that we've noticed is that human brain inside that skull has grown as well. It actually ends up being that the size of the brain grew even at the faster rate than the size of the skull. So that gives us a little bit of a challenge how we as a species and changes of the over the long historical time scale have dealt with that. So one of the things that we end up seeing that has happened over time is that our anterior portion of the brain, so really the frontal lobe, it's grown much bigger than it was earlier in the history. But also, like I said, the brain grew, and especially the surface area of the brain. And now we're faced with a challenge. How do we pack more surface area within the skull that even though the skull grew in size, how did we fit even more surface area in there? And I think that one of the easiest way to think about it is that if we're comparing the surface area of this post-it note and the surface area of this regular sheet of paper, obviously this regular sheet of paper needs a much larger space to fit it in. It's kind of an analog of the amount of surface area that happened with our brains. But skull, even though it grew, it didn't grow that much. So how can we fit all this surface area there? And one of the things that we end up seeing is that we ended up developing folds in our brain, on the surface of our brain. So you end up seeing that the early brain was much smoother from the surface, but in order to facilitate that large surface area, we ended up developing all these different kinds of folds. So that's one thing to notice. Another thing that I want to talk about real quickly is that what happens during a lifetime of an individual. Let's talk about that a little bit. So brain is one of the first systems to start developing during early development at week 2 or so. We start to see already some primitive structures, but really the development rate is really fast during the prenatal development before the birth and also during the early years of the life. Then the development rate slows down a little bit, but notice that we never really stop developing. I think that there's a little bit of sometimes misconception that you're just stuck with a brain. There is actually constant changes that happen. So let's have a look of a little bit of the early development, even though I don't really test you much about that. I think it's interesting just to kind of have an appreciation and understanding of what we're going to be talking about. So when we look at how human brain develops, we really start as this kind of a neural tube in early development, just nothing but a tube of nervous tissue. But this tube of nervous tissue then goes through various different changes as we develop prenatally, and after that we start to develop different kinds of folds in that tube. And there's also thickening of certain areas, and we end up seeing distinct regions also developing um, that take place there. And really, when we look at the adult brain, there's, I guess, 4 main regions that we could talk about. So we can talk about the cerebral hemispheres, these big parts of what we see outside and often think when we talk about the brain. So we'll spend some time today talking about cerebral hemispheres. In addition to that, we have structures buried deep inside, kind of middle of the brain, inside the brain structure. These are going to be our diencephalon, and we'll have a look at a few important parts of the diencephalon later on. We also will talk about the brainstem, which is really made of, again, a few specific portions, and it's the first part of the brain to develop in terms of your development during a lifetime, but also evolutionarily. So this holds some of the most important functions, such as life-sustaining regulatory functions. I think of that not just on an individual development, but also evolutionary development, but also in a sense that brainstem is very well protected. We want to protect those vital functions. And finally, we'll have a quick look of the cerebellum, which is often sometimes referred as a little brain at the back of the structures that we'll see. So we'll go through each of these very briefly today and in more detail in your full lecture videos. But before we do so, let's have a look of a concept of the gray and white matter. So some of the brain tissue will appear kind of more whitish or palish in color, and that's because this kind of a brain tissue has a lot of myelin around it. So you remember last time when we talked about a typical neuron, we saw that the axon of the neuron could be surrounded by these fatty blobs. That was the formation of the myelin, and myelin helped to speed up the passage of messages, kind of also served a little bit of protective functions, and so on and so on. But in addition to this myelinated tissue, which is really, you could think of tissue that helps to pass messages from one part of the brain to another, we're going to have also kind of a little darker tissue. We call this gray matter. This is tissue that does not have myelin around it, and this is mainly the cell body. So this is where the activities the processing of the information happens. So one good way to think about this is that the gray matter is the finer processing, whereas white matter is really passaging messages. It is a bit of an oversimplification, but that might give you a rough idea. I don't know how many of you have read Sherlock Holmes books or watched the movies, but there's often a reference with Sherlock Holmes that when he's solving a crime, he's gonna dig into the gray matter that he's doing. That refers that he's thinking about the crime or the problem that he's facing. So really, this gray matter is where a lot of that processing happens. I don't know if that helps you to remember the concept that way. So where do we find the gray and white matter? Well, let's start by looking at the brain. Here we have a cross-sectional view of the brain, and we'll end up seeing that the cortex of the brain is largely made of the gray matter. So that's where we find most of the nerve cell bodies. You could think of it that this is where the processing of the neural impulses of the information happens at the cortex of the brain, whereas the center of the brain largely is more of a white matter. This is where those myelinated axons are very predominant. So this is just kind of more of passing messages along. Obviously, this is going to be an oversimplification, and we're going to have specific parts within the brain as well where different things happen, but just kind of to have a broad bird's eye view. What's interesting is that when we look at the spinal cord that continues as an extension of the brain, we end up seeing that this order of the gray matter and white matter is reversed there. So the gray matter is found kind of more deeper in the spinal cord. That's where the nerve cell bodies are located. Whereas the outer part of the spinal cord is going to be the white matter, the passage of the messages. Obviously, if you go into more detail in your studies and study neuroscience like I did at the university, you end up seeing that even within the spinal cord at different level, there's going to be all these blobs of different structures and different colors and different passages ways of the nerve impulses in spinal cord are much more complex. So you'll get to learn about that if you're interested in it. So let's have a look at the next concept that I wanted to emphasize and show. We're going to see that inside the brain, there's also going to be empty spaces that are fluid filled. So all of these connect to each other, and the fluid that we end up finding inside these empty spaces is going to be our CSF, so cerebrospinal fluid. These are what the structures that we're seeing, the empty spaces within the brain structures, within the brain itself, are what we know as ventricles. And if you think of back what we discussed last week, we actually see that these ventricles are lined by the ependymal cells. So those boundary forming cells that we talked about, the glial cells that support the nervous tissue. So we're seeing that all of these little concepts that we talk about are also part of the bigger concepts. So again, more information about the ventricles, you can refer back to the full lecture materials, but I think this is good for our review purposes. Let's have a look of the cerebral hemispheres a little bit. Approximately 83% of your brain mass is formed by the cerebral hemispheres. And there's some terminology that I want to make sure that we're familiar with as we're talking about these structures. So like I showed before, during evolution, the amount of surface area of the brain grew, and that's why we ended up developing all these folds. So a couple of words when we're talking about the gyri, we're talking about these ridges or these bumps. When we're talking about sulci, we're talking about these grooves or depressions between these bumps. And another word that we see sometimes is going to be our fissures. Fissures are just very deep or very prominent grooves. So there's going to be some very specific ones that we'll notice and we'll name. And let's start with the two of the most important fissures first. So we're going to have our longitudinal fissure, which separates the two hemispheres. So you're going to have your left and you're going to have your right hemisphere separated by our longitudinal fissure, as we're seeing here. You're also going to have another really important fissure that separates our cerebellum and cerebrum. So separating the little brain from the rest of the cerebral hemispheres. That's going to be our transverse cerebral fissure. Transverse cerebral fissure. There are going to be other ones that we'll see as well. And what I'm going to do in just a moment is to divide the brain and the cerebral hemispheres into different portions that are separated by very specific depressions. So let's start with those depressions, separating the brain into different parts, and then talk about those different parts. So we already saw our longitudinal fissure, which you wouldn't see on this picture because that was separating the right and left cerebral hemisphere. But one of the structures that you're going to see is the central sulcus. Central sulcus is going to separate our frontal and our parietal lobes. So there's going to be very prominent depression running in here. And that was known as our central sulcus. What else can we see? We already talked about the transverse cerebral fissure. We're also gonna see our lateral sulcus. Lateral sulcus separates our temporal lobe from the other structures. So it's kind of a little bit separate structure, a little bit distanced from the other parts. Another one that I think is an interesting one is our parieto-occipital sulcus, which separates our occipital lobe from our parietal lobe. So there's also this depression that we can see there. And these are found on all people, even though we're unique, even though we're different, these structures you would end up seeing. So let's take all of these depressions between different parts of the cerebral hemispheres and use them to guide us as we're separating different lobes of the cerebral hemispheres. So we're going to see really 4 lobes, and then I'll add a little bit more detail to that. So let's start with our frontal lobe. So frontal lobe is the most anterior portion of the cerebral hemispheres. Remember, this is the part that grew over the evolution to be bigger and more dominant. So we didn't see as large frontal lobe in early humans. When we compare humans to other animals, one of the things that's very characteristic of human nervous system and central nervous system is that we have very, very large frontal lobes. Frontal lobes are really where the thinking, planning, a lot of executive functions take place. So you can think of it in a way that if you compare us to other animals, we do quite a lot of this kind of processing that has to do with executive planning, executive functions, having hopes, dreams, more of a analytical thinking in that sense. So frontal lobe is very, very prominent for us. At the posterior portion of the frontal lobe that we saw when we were talking about that separated this groove, so that separated our parietal lobe and frontal lobe. So at the back of the frontal lobe, we're also seeing our motor areas of the brain. So where these different regions of this cortex of the cerebral hemisphere, this part of the cerebral hemisphere, the frontal part, you can actually locate motor functions along this structure. Moving onwards, let's have a look of the parietal lobe. On the parietal lobe, again, remember the bones of the skulls. This is where it's going to come handy, all the effort that you put in to learn those. This is where a lot of sensory functions are located, and they actually map opposite to the motor functions on the other side here, forming kind of a really good match on both sides. So a lot of sensory functions from this lobe. Then we see our temporal lobe, which was kind of located here on the side. This is especially where we see things like language, hearing, speech, production, processing happening. And finally, our occipital lobe, which is really reserved for vision and processing visual information. Deep into these structures, you're also going to have, if you kind of separate the temporal lobe a little bit, you're going to see a deep structure known as insula as well. So I just wanted to put it out there to highlight. So you might ask me that how do we know these different functions that are dedicated to different lobes of the cerebral hemispheres? Well, a lot of that information is from the study of injuries. So we saw earlier on at the very beginning, last week, we talked about the case of the man who mistook his wife for a hat. We talked about the case of Phineas Gage. So we're seeing that a lot of the time, the information that we've gained about these different regions has been through pathological processes, has been through injuries, and then studying the changes that we see on the individual, maybe later on analyzing after they're passing their brain. We even can do that today, imaging while they're still alive, but if there's an injury, that what is impacted on them. The other thing that we do a lot is brain imaging, which is my former life and my former job. So we end up seeing that as you're performing different kinds of tasks and tasks that would be very characteristics for different parts of the brain, those parts of the brain that are active, we will be able to tell that apart with the brain imaging. So where there's increased blood flow to those areas where there's higher oxygen use. So we can again locate functions through functional brain imaging, for example, on these different regions. We can take this even further and we can identify not just different lobes of the brain, but we can identify very particular parts. So two that I've highlighted here are our Broca's area, named after the person. This is really specified for speech production, and Wernicke's area, which is more of about to do with speech comprehension. Like I said earlier on, we have also the motor and sensory regions that we see there. A lot of the time, my students are really curious about talking about when we're talking about cerebral hemispheres and left and right hemisphere, like we saw earlier on, is this concept of lateralization of these cortical functions. Sometimes people get called left-brained or right-brained. There's certain different functions that are more dominant on different sides, but there's a little bit of debate whether this is really proven and whether this is more of a layman's science, if you wish, in that sense. In the interest of time, I'm just going to keep going on, and let's have a look at two more structures. We're going to start by talking about the diencephalon, this part where we're ending up seeing the parts inside the brain embedded deep in there. After we've covered those parts, we'll also have a look at the brainstem, this part of the brain that comes out and then turns into the spinal cord later on. But let's make a start with this diencephalon and notice a couple of structures from there. So the structures that I want us to spend a little bit of time are our thalamus. We'll talk about that, and we'll also talk about hypothalamus, which is located underneath the thalamus, like the name tells us. We'll also talk a little bit about the epithalamus as well, which I think is going to be interesting. So let's start with our thalamus. I'm gonna just use the same picture just to make it a little easier for us to follow along. This is kind of the egg-shaped blob that we end up finding on both sides, and there's fibers that communicate with the cerebral cortex. You can really think of this as kind of a relay station for the information coming into the cortex, sorting and editing and relaying information. A lot of things like emotions, visual functions, direct motor functions, and also the integration of memory and sensory information to form kind of these whole experiences. So those are some of the things that go with the thalamus. Let's have a look at the hypothalamus. So like I said, a name literally tells us that it's below the thalamus. And things like, for example, the smell relay station would be found there. It's noteworthy to notice that hypothalamus connects to the pituitary gland, and we'll talk about pituitary gland in a great extent when we talk about endocrine system later on. In terms of the hypothalamus, this is really regulation center for many functions that have to do with homeostasis in the autonomic nervous system, such as blood pressure, heart rate, digestive tract functions, pupil size, really physical responses to emotions, regulation of your body temperature, sweating, shivering, kind of that go with that. Also things like feeling hunger, feeling full, feeling thirsty, sleep-wake cycle, we'll kind of touch upon that when we talk about endocrine system. Uh, Really, the most important role, I think, with the hypothalamus is that control of the endocrine system via the pituitary gland. So that's going to be kind of an important in that sense. When it comes to the epithalamus, we've got a couple of structures there. We'll see our pineal gland. Pineal gland produces a hormone known as melatonin, which regulates your sleep-wake cycle. Again, we'll get to talk a lot more about that when we get to the endocrine system as the first chapter of your BIO202 course. I think we're ready to move on to talk a little bit about the brainstem. So with the brainstem, we've got three different important parts. We've got our midbrain, which is the kind of the part inside the brain. We've got our pons, the kind of the first bump that we see. And then we've got our medulla oblongata, often called just medulla, the second bump that we see. This is really like spinal cord, but really contains the vital functions closer to the brain. What would I say about the midbrain? I think that really a relay station is a good description of that. With the pons, some of the functions would be, for example, regulation of the breathing rhythm. With the medulla, I think that we see things like equilibrium relay of that, reflexes, heart rate, blood vessel diameter regulation, breathing regulation, things like responses like hiccups, vomiting, swallowing, coughing, sneezing are especially relevant to the medulla. Moving onwards and upwards again, let's have a look of the cerebellum real quickly. This is our often known as our little brain, about 11% of the brain mass. And this is where really the movement and balances are controlled. If we take the cerebellum and cut it in half, we'll actually see inside there these myelinated tracts that are very prominent, and we see them. These are sometimes referred in language of anatomy as the arbor vitae, which really translates to the tree of life. So very, very important tracts passing those messages that we see there. We could talk a little bit about spinal cord, but I think I'm going to save that for the coming weeks. Like I said, a lot of these weeks that we're having a look have some overlap, so we'll get to visit that in a little bit more detail as we carry onwards. I do quickly want to show you a little bit about what comes to the homework for this week. So let me just jump out of sharing my screen here. And what we're going to do instead is, if I get my screen sharing to end, I'm going to share the homeworks for this week so you have an idea of what to expect with those. So we're going to be working this week on 2 assignments. We're going to be looking at our chapter 14 quiz and our chapter 14 practice anatomy. And let's get these opened up for us to have an appreciation of what kind of things might come up. So let me share this screen with you. So for this week's homework, what I'm looking for in the chapter 14 is going to be a quiz that's worth of 5 points. It's not a super long quiz, but I think it's a worthwhile quiz and activity to do. So some of the examples of the types of questions are, again, these drop and drag or drag and drop, where we're moving different structures and classifying whether they fall within the gray matter or whether they're more of the white matter. And you'll get to learn about all of these more detailed structures as you review the full lecture videos. What else do we get to see? We get to do also indicating these functions that we talked about, the cortical lateralization, whether things are more of the left hemisphere or right hemisphere. So these functions, you get to do a little bit of identifying that. We get to complete some sentences that describe the things that we'll get to talk about when we're doing more detail about the central nervous system. We'll also see, in addition to even further statements, question types like our traditional multiple choice questions. For example, on this review, we didn't get to talk too much about the corpus callosum. Corpus callosum is the part right in the middle of the brain that really is the pathway that connects the 2 hemispheres together. So corpus callosum is there. We've done in the past, early days, when people had really severe epileptic seizures, we could cut their corpus callosum, hoping that it would stop the epileptic seizures from spreading throughout the brain. But really disrupting this connection between the 2 hemispheres had pretty dramatic consequences for the patients in general as well, and their nervous system activities. We'll also get to do a little more detail of these different regions of the brain, identify structures, identify functions that are associated with them, and more multiple choice questions. So that's something that you'll get to see as we're working through the homework. I also want to show you really quickly the chapter 14 practice anatomy. So let's have a look of that as well. And again, this is going to be word of 5 points, not a huge amount of questions. So this should be kind of a nicer week in sense. We're identifying different structures that we've talked about, like our cerebral hemispheres, the left and right sides of the cerebrum. We're seeing the different lobes separated by different kinds of fissures, so such as the longitudinal fissure dividing the cerebrum into left and right sides. We're also seeing, for example, our central sulcus that separated our brain on this deep groove here. We're seeing our frontal lobe, the anterior portion of the cerebrum, and we're seeing the parietal lobe, kind of superior to the occipital lobe if you orientate yourself with this picture where we're looking down on someone's brain, front here, back here, anterior, posterior part. Occipital lobe, which was the posterior portion of the cerebrum. So you really get to practice all of this stuff that we've talked about. Some other structures, again, we're seeing our central sulcus that separated the frontal and parietal lobe. We're seeing durae, which were those ridges or folds within the cerebral cortex. We're seeing the cerebrum, the largest area of the brain. Lateral sulcus that separated really the temporal lobe. that we're seeing here. We're seeing brain stem, we're seeing cerebellum, we're seeing spinal cord. So a lot of things that you have already learned about, kind of a way to guide you. I've got some real pictures of actual structures as well, actual human brain. So you get to identify those different lobes, those different structures that we talked about. Some more diagrams here. We're seeing the little brain, the cerebellum. We're seeing nicely that arbor vitae, that is white, myelinated pathways, different ventricles. We'll get to talk about structures of the brain stem. Again, notice on this one that anterior is on this side, posterior is on that side. What else? I have more diagrammatic colored diagrams that you can look at, different lobes, specific areas, like we're seeing the Broca's area that was involved with the specific functions to do with speech production, for example, and so on and so on. We're seeing that motor cortex and some of the sensory cortex. So we're seeing those two kind of correlating, mirroring each other, and so on and so on. I think that that gives us a good idea of the homework that we're going to be completing this week. Let's have a look of a little bit more, just to wrap up where we finished our presentation for today. So I think that we had a good review of we checked in, we looked at the homework, we talked about the key aspects, really focusing on the chapter 14. We had a look of the homework, and that brings us to the very end, which is really for me to say that I want you to know, again, even though this is an asynchronous class where you can work at your own pace, it doesn't mean that you don't have support for you. So all you have to do is reach out for me if there's anything that I can do to assist you. We are approaching the finish line. There's not that much more to go. So let's cross that strong and come to a great ending of the semester. I really appreciate you being part of this class, and I look forward to another great week of learning with you. Until I see you next time, have a good week, and I look forward to working with you on the Canvas where we'll be interacting a little bit. Thank you for this. Bye.